This video will cover the process of antigen processing and presentation by the major histocompatibility complex, or MHC. Our video will cover an introduction to MHC, the cytosolic pathway, the endocytic pathway, and cross-presentation. B cells can directly detect microorganisms and their antigens. T cells, however, require antigens to be presented by MHC molecules on cell surfaces. This allows for immune surveillance against potential invaders like bacteria, fungi, or parasites. MHC molecules are protein complexes that present peptides to T cells, allowing T cells to monitor what's happening inside a cell. If a cell is infected, viral protein fragments are displayed on MHC molecules, enabling the immune system to identify and respond to the infection. There are two classes of MHC. MHC Class I presents antigens from intracellular pathogens, while MHC Class II presents antigens from extracellular pathogens. Antigen-presenting cells have both, but nucleated cells only express MHC Class I. MHC compatibility is crucial in grafting. For instance, in these mouse models, successful skin grafts depend on matching MHC haplotypes between donor and recipient to prevent rejection. Antigen processing is essential for T cell recognition. As shown, fixation methods like paraformaldehyde or glutaraldehyde affect whether an antigen-presenting cell can activate a T helper cell. Antigen processing and presentation involve both the endocytic and cytosolic pathways. These pathways differ in their peptide source, the MHC molecules involved, the T cells they present to, and the types of pathogens processed. In the cytosolic pathway, the proteasome degrades large antigenic proteins into smaller peptides, which are then presented by MHC Class I immunoproteasomes, induced by interferons, offer faster and more effective processing for MHCI binding. This diagram illustrates the intricate process of peptide binding to MHC Class I molecules within the RER lumen, involving various chaperones and enzymes to ensure proper assembly and presentation. Once MHCI presents a cognate antigen on the cell surface, it activates cytotoxic T cells. This activation initiates the perforin granzyme pathway, leading to the apoptosis, or programmed cell death, of the infected cell. MHC Class II presentation is handled by antigen-presenting cells like dendritic cells, macrophages, and B cells. These cells efficiently take up extracellular material and process antigens for presentation. Extracellular antigens enter the cell through endocytosis, forming endosomes or phagosomes. These then fuse with lysosomes, where degradative enzymes break down the material into peptide fragments. MHC Class II molecules are assembled in the rough ER. An invariant chain, or 2, binds to MHC2, preventing premature antigen binding. HLA-DM then facilitates the exchange of C-clip with antigenic peptides. The MHC Class II clip complex fuses with an endolysosome, forming the MIIC. Here, HLA-DM releases C-clip, allowing degraded peptides to bind to MHC Class II. The complex then moves to the cell surface for presentation. The CD4 molecule on T cells interacts with MHC Class II, enabling the T cell receptor to survey the presented peptide. If the TCR specifically recognizes the peptide, the T cell becomes activated. Activated CD4 T cells can enhance immune responses. For example, they help macrophages kill phagocytosed bacteria by secreting cytokines, or assist B cells in producing antibodies when they present cognate antigens. Cross-presentation allows antigen-presenting cells to load internalized antigens onto MHCI molecules. This is vital for immune responses against viruses and tumors, preventing accidental activation of T cells. Here are the references used in this video for further reading and research. Thank you for watching this video on antigen processing and presentation by MHC.